My chosen topic applies to biotechnology and forensics. It is about researching the use of microbial biosensors for detecting traces of ignitable liquids in fire debris. The outline I have planned for this paper is formatted as a research proposal. I plan to organize it in the sections I have written down here. And if anyone doing a review for me has done undergraduate research or is more familiar with research proposals, I'd really appreciate any formatting tips for how I should organize the paper. So my research thesis is pretty straightforward. By studying and applying microbial biosensors, the aim is to enhance the techniques for identification of common ignitable liquids used in fire investigation cases. The focus is on their application in forensic investigations and for substance identification. For some background information, currently microbial biosensors are being utilized for detecting unactivated landmines buried underground. In my first reference link, I linked one study being conducted for this purpose. I found a lot more than that, but I haven't really gone over them in more detail, so I did not add them in this presentation. I found another research article. It's my second reference listed. It's about detecting ethanol in alcohol. Ethanol is pretty common and super flammable. So that is like a step in the right direction of what I wanted to base this research proposal on, but there's still a need to expand the scope to cover like a broader range of ignitable liquids, but specifically the ones commonly encountered in forensic investigations. Some other uses of microbial biosensors include monitoring and detecting pollutants in the environment, the detection of heavy metals in soil or water, and detecting gases such as methane, carbon dioxide, or hydrogen. The uses aren't limited to just forensic applications. Some stuff I found to be missing from the resources I've ended up finding so far are any sort of focused study on these biosensors to detect common ignitable liquids. So substances like gasoline, diesel, propane, acetone, paint thinner, lighter fluid, etc. Just flammable, common things that you could find in your house. Some current techniques for detecting ignitable liquids are included in this list that I have here. So the most commonly used method is gas chromatography mass spectrometry. This technique involves separating a mixture into its individual substances by heating it. In fire investigations, the GCMS identifies any constituents found present in the sample mixture, and this chemical signature is used to determine the identity of the accelerant. Other methods that are currently applied to identify accelerants include gas phase sampling, also known as headspace sampling, Fourier transform infrared spectroscopy, and photoionization detectors. However, these methods do have limitations when analyzing fire debris. For example, some of these methods may not be able to detect enough characteristic compounds to identify accelerants more specifically, especially when very small amounts of sample are able to be collected for analysis. This commonly happens in fire investigation cases. So an explanation of how microbial biosensors work is a microbial biosensor is a tool that combines microorganisms with a transducer to generate a signal indicating a measurable concentration of specific substances. Some examples of biosensors include enzymes, microorganisms, DNA, and lipids. For the research I'm writing this proposal for, it would utilize microorganisms as the biosensors. So to explain it in a little bit more detail, so how a biosensor works is that an analyte will interact with the biological recognition element in the biological receptor. The biological response from this interaction will be converted into a detectable physiochemical signal through the transducer. The signal will then be amplified and converted into a measurable numeric amount through a microelectric processor. In the context of this proposed research topic, Microbial sensors have been effectively utilized in detecting landmines and explosive residue, showcasing their potential for identifying volatile substances. Their ability to interact with specific volatile compounds makes them a good potential candidate for ignitable fluids identification and forensic investigations. So some potential negative impacts that I considered. So a challenge that could be faced is the cross-reactivity with other substances could lead to false positives or misinterpretation of results. Another challenge is that the performance of microbial biosensors may be influenced by environmental factors like temperature, humidity, or presence of other microorganisms. These variations may affect the reliability and consistency of results in different fire scenes. For ethical considerations, an ethical concern to consider would be about maintaining transparency in the use of microbial biosensors. Public acceptance and understanding of these technologies could impact their integration into forensic practices, so it would be best to be as clear as possible with the processes and the methods and potential implications of using this technology in forensics. For socioeconomic impacts, the initial costs and accessibility of microbial biosensor technology may pose challenges, particularly for smaller forensic laboratories or those in resource-constrained regions. Addressing these issues is essential to ensure broader adoption and equal access for this technology. The intended outcome of this experiment will be to contribute to improving the best practices in ignitable liquid identification and detection. The intention is for the knowledge gained to be applied in forensic investigations and potentially proving to be a more effective detector for fire accelerants than the currently utilized methods. That is the intention of researching. In summary, the research proposal for my paper will address studying and applying microbial biosensors for the identification of common ignitable liquids used in fire investigation cases, and to consider it as a more enhanced detection technique than what is currently used. While there are still some challenges that should be addressed in order to increase the accuracy and specificity of the microbial biosensors, they are a promising candidate for an improved accelerant detection technique. Public acceptance and understanding of these technologies can affect their acceptance in being used in forensic investigations. There is a potential for socioeconomic impact regarding cost and accessibility, but with adequate research and planning, I believe these issues can be easily mitigated. The envisioned outcome of this research is not only to advance our understanding of the use of microbial biosensors in ignitable liquid detection, but also to actively enhance best practices in forensic investigations. The goal is that the knowledge gained from this research will establish microbial biosensors as a more effective and reliable detector for fire accelerants. These are some of the references that I used just in this presentation. I have many more. Those will be just for the paper though, because I haven't gone over them in more detail. And that is it. Thank you.